For a long time, microbes have been synonymous with plagues, diseases, and epidemics. But in recent years, research from around the globe has revealed that microbes are in fact essential to all forms of life, plants, animals, and of course to us humans. The bacteria inside us are useful for breathing, eating, and growth. They can even influence our behavior. And sometimes, they can even change the destiny of a species.
Looking into the microbial world is as transcending as looking through a telescope to the stars and thinking about our place in the universe.
We can't keep them, but with the observation tools available, we can reveal their existence.
This is a very novel time. A lot of people feel that we have not been at this kind of inflection point since Darwin.
The study of bacteria is revolutionizing all we know about how living things function, and could possibly revolutionize medicine. We can't see them, but they number in the billions. Their presence is beyond question. We are immersed in a sea of bacteria, in water, on the leaves of trees, in the air we breathe. They inhabit every tiny corner of the planet. Here in Amsterdam Zoo, a contemporary museum called Micropia was created to make microbes visible. Bacteria first appeared 3.5 billion years ago, making them one of the very first forms of life on Earth. They're also among the most basic, consisting of a single cell without a nucleus, with a few DNA molecules floating around inside. Invisible to the naked eye, bacteria exist in all shapes, round, rod-shaped, or with filaments. Since their advent, they've multiplied to colonize the entire surface of the globe. They're present on the most everyday of objects. Bacteria are everywhere, absolutely everywhere. They form a world of their own. And now we're finally beginning to understand the complexity of this world.
The goal was always, after you leave Micropia, you can never ever look the same way at yourself as before. You are part of nature, and you're not just this individual, but you are full of other organisms which make us do what we can do. And this is the start of a new thinking, of a new era, and I think it's like Steve Jobs coming out of his garage with the first computer. We will see in the future a big explosion in any field.
Over the past 10 years, microbe research has taken off around the globe, engaging teams of scientists in many different countries. All areas of life sciences are involved: biology, genetics, medicine, behavioral sciences, and even the science of evolution.
For the last 100 years, really, biology has focused largely on genetics and DNA inside the nucleus of animal and plant cells. What we're interested in is asking how important is microbiome in affecting these questions, in driving the definition of who we are as a biological entity.
Here in Nashville, Seth Bordenstein works at Vanderbilt University. His research on the mechanisms of evolution may possibly revolutionize our conception of the human species.
The goal of our work is to understand who we are as a species. What does it mean to be Homo sapiens? And who we are as animal-microbe associations. Because we do not live alone. We live with trillions of other microbial cells and thousands of other species.
Like many of his colleagues, Seth Bordenstein studies insect models, and his research has led him to redefine what constitutes a living being.
And what many people didn't know is that there is no individual, but the animal and plant is an assemblage of individuals. It could be called an ecosystem. It could be called an assemblage, a community.
Ecosystem, assemblage, community. Our traditional notions of animals and plants are being completely overhauled by the study of microbes, because the whole animate world lives with bacteria, and sometimes even thanks to them. But how does this strange coexistence function? At the University of Tsukuba in Japan, the research of Professor Takuma Fukatsu vividly shows how bacteria can be indispensable to the survival of a living being. A delicate operation is taking place, the dissection of an aphid to remove its digestive system. Professor Fukatsu is searching here for a very specific type of cell.
You can see in the aphid body, there is a special cell called bacteriocyte. And inside, there are so many particles. They are all bacterial cells. And you can see how many bacteria within a single cell. That is very amazing.
Each of these red dots is a living microorganism, autonomous with its own DNA, and which has found what it needs to feed and proliferate inside the aphid's intestinal cell. Invisible to the naked eye, this microbe, dubbed Buchnera, measures just 3 microns, 3/1000ths of a millimeter. In the aphid's intestine, there are between 5 and 6 million of them.
Why so many bacteria within the cell, within the aphid body? The reason is this bacteria provides the aphid with essential nutrients called essential amino acid. So if removing this bacteria by using antibiotic from the insect, the aphid suck the plant juice, but they cannot grow because they cannot synthesize proteins without this bacteria. So this bacteria is really nutritionally very important.
Without this bacteria, aphids would be unable to absorb the sap from plants and, as a result, be unable to grow and develop. The aphid's relationship with the microbes is vital.
Their association was established 100 million years ago, the age of dinosaur. And so these two organisms, aphid and Buchnera, are almost a single biological entity now.
Scientists call this working relationship between the bacteria and its host symbiosis. And sometimes such collaboration can be extremely close. In Italy, off the coast of the island of Elba, a small marine worm and its bacteria have surprised researchers. This little worm, Olavius, has a remarkable feature. It has no mouth and no anus.
Perfect.
We saw a lot floating while taking the samples, so I hope there are a lot.
Yeah, no, that's a
here.
good sign. Whenever you talk to someone, whether in science or not in science, and you tell them, I'm working with worms that don't have a mouth, that don't have a gut, they don't have kidneys, and everyone is like, how do they live then? And that's really fascinating that nature has evolved so efficiently that they are able to exist without all of these and are still able to thrive. So yeah, I think it's quite fascinating.
How has this little worm been able to survive without a digestive system? And why did this extreme and surprising system develop? It's a double enigma because the environment in which the Olavius lives is so hostile. The worm thrives in this sandy sediment. It's not only very poor nutritionally, but is packed with substances that most creatures would find poisonous. Substances such as carbon monoxide and hydrogen sulfide. It's thanks to images taken with an electron microscope that Olavius's secret could be revealed. Each of these small formations on the surface of its skin is actually a bacteria that converts toxic substances, compounds of carbon and sulfur, into nutrients for Olavius.
It's similar to photosynthesis, so the same way plants produce food, so they are able to use carbon dioxide, they're able to use water, and they're able to use energy from the light to make food for themselves. And in a similar manner, the bacteria convert this inorganic carbon into organic carbon, and that the worms can use for energy, to grow, to gain some length, some tissue. So that's their nutrition.
The microbes living on the worm's skin function as an organ. It's the equivalent of our intestine. Humans can no longer view themselves as autonomous entities. Biologists now consider all the bacteria that colonize the body as an organ in its own right, just like our lungs, liver, and heart. This organ has a name, the microbiota. Its functions are innumerable: the immune system, digestion, development of the coagulation system, synthesis of certain vitamins, and the protection of our skin tissue. In an adult human, the microbiota can weigh up to 2 kilos in total. But how do we acquire these microorganisms? Are we merely contaminated by our environment? Or are the bacteria transmitted from parents to their offspring? The surprising behavior of a small Simicidae studied by Takema Fukatsu reveals some clues.
Very strange animal it is. Yeah.
This bug lays its eggs in a kind of jelly that totally encapsulates them. When the eggs hatch in midwinter, the larvae feed on the jelly until the arrival of spring. But this is not its only use.
One after another, 20 or 30 eggs are laid in a row on the bark. The mother produces a jelly by their ovary, jelly-covered egg. The symbiont is also excreted and mixed up with the jelly, and the jelly-egg complex come out. And in this way, the bacteria is transmitted from the mother to offspring by way of the jelly.
So, generation after generation, the essential bacteria are transmitted to the bug through this maternal jelly.
That is the only known from this group of insects. But anyway, all animals are dependent on the microbes in some way. And yes, of course, in human, the baby acquire their gut microbe from his or her mother.
Just as with insects, a crucial process plays out at the birth of humans and other mammals. It is at this precise moment that you acquire and develop your microbiota. After spending 9 months protected from all external bacteria, babies enter into contact with the microbes that will be indispensable for the rest of their lives.
So at birth, it's a shock. The infant will be contaminated by its mother. The mother transmits the microbes present in her vagina, anus, and on her skin. And therefore, it's an invasion, so to speak. Then the baby will feed on breast milk, and this milk also contains certain microbes. Then when it goes on to solid food, many new bacteria colonize the baby, and these new bacteria are those that it will more or less keep until the end of its life.
And from birth, the bacterial invasion will trigger the creation of another biological system essential to survival, the immune system. The organs of this system, bone marrow, spleen, or lymph nodes, produce lymphocytes. These cells and their antibodies are responsible for controlling the activity of microbes and, if necessary, for neutralizing them. What's surprising is that the immune system needs microbes to become operational. This is why a baby born by cesarean section may have a less effective immune system than a child born naturally.
It's a little like sports. To be effective, you have to train all the time. An immune system that's not trained, that's not in constant contact with bacteria, is much less efficient.
As you develop, you come into contact with more and more bacteria. Little by little, your immune system becomes stronger. The bacterial population you're hosting grows to gigantic proportions. Adult humans have as many bacteria as they have cells. When two people kiss for 10 seconds, nearly 80 million bacteria of 700 different species mingle and are exchanged. This is how our internal ecosystem is modified, and usually that's a positive thing.
There are so many beneficial microbes. So there are some microbes may do bad things, some do good things, some do both, and most of them we don't know.
For over a century, microbes were regarded as disease carriers to be fought with antibiotics and antiseptics. Such medical thinking is now widely questioned.
We tend to treat bacterial infections with antibiotics. However, an antibiotic cannot target a specific bacteria. It will kill the other bacteria responsible for the disease, but many other bacteria as well.
That's why doctors are now cautioning against the systematic use of antibiotics. And it's estimated that less than 1% of the bacteria in our bodies are harmful. Compared to the thousands of microbes that are useful to us, only a few dozen can prove dangerous.
But probably the accurate way to think about microbes is they are just microbes. Sometimes they're bad, sometimes they're good. And this depends on a myriad of variables. including what types of genes does that microbe have? What types of genes does that human have? What's the environmental pressures? And it really takes all three of those kinds of variables to determine whether a microbe is going to be good or bad.
Microbes are thus essential factors in our health. In our intestines alone, researchers have found 19,000 different functions that are provided by over 160 species of bacteria. This is what is commonly called our intestinal flora. The intestinal microbiota has become a major focus of medical interest. In the southern German city of Heidelberg, the team of Dr. Peer Bork, a pioneer in the exploration of the intestinal microbiota, is leading a worldwide study. They're collecting human excrement from around the globe to study its microbial composition. Developed on the web, the project has already got samples from a lot of volunteers. And scientists studying the microbiota collection gathered in Heidelberg quickly discovered something startling.
Ja, als wir 2011 hatten wir eigentlich keine Erwartung, weil keiner hat je reingeguckt.
When we started in 2011, we didn't really have any expectations because no one had ever worked on this. But we might, for example, have assumed that no two cases would be the same, or that there would be links between the bacteria and the different eating habits in various countries. The Japanese eat a lot of fish, and that has a certain composition. The French drink wine, and that would be reflected. And what surprised us was that food, geography, and also gender and age apparently have no impact.
By comparing samples from different continents, the researchers observed that human intestinal microbiota could be classified in three main categories that they have called enterotypes. The three enterotypes are like three large intestinal flora families. What type we are does not depend on our environment, our sex, our age, or even on our diet. In fact, scientists still do not know what determines our gut type. And yet this is what dictates how we digest our food or react to a drug. Research in this field is still in its infancy, and yet Professor Bork's team has already established correlations between changes in intestinal microbiota and diseases such as colon cancer or obesity. These findings sometimes go beyond mere diagnosis and offer new prospects for treatment.